Great. Okay, so my name is Matt. I'm a data scientist at the International Brain Lab. And now we're going to switch gears a little bit and talk about the functional analysis of neuropixels data. Uh, I guess there's a lot to be said about functional analysis of neuropixels data. This is very complex, interesting data, and there's a lot to do with it. So today we're actually just going to focus on some very simple things. And tomorrow, actually, there'll be a couple of talks that dive into the IBL data more specifically and some of the analyses that we've done there. So to start off, I'm going to talk about the actual International Brain Lab brain-wide map data set, because now we're talking about characterizing neural activity in the context of some task or some behavior. So I want to talk about this particular example first. So we have a standardized decision-making task that we've developed in the IBL, and I'll talk more about the details of this task on the next slide. But I want to say that in this data set, which is publicly available, and some of which you all have already looked at, I think, we have almost 700 neuropixels insertions across 139 mice, and these have all been aligned to a common mouse brain. And so this is really taking advantage of a lot of the tools that you have seen already. And so here now you can see sort of a visualization of this entire data set. Again, this is all of the trials, all of the mice kind of, put into one single animation. But you can really see how the neural activity changes as a stimulus is presented and the animal indicates its decision, and then they get some sort of feedback. So this is a really big, complex data set. And I should also say there's a lot of behavioral variables that we're recording as well. The wheel movement, and we're collecting video, so there's some sort of pose tracking going on as well. And so we really need to come up with ways of kind of visualizing this data and looking into it. So this is going to be the first step of this understanding the functional properties of these cells. And so I'm going to talk to you in a couple slides about this interactive data visualization website that's been developed by the IBL. So first I want to talk about this decision-making task in a little bit more detail. And again, this will come in handy for the talks tomorrow as well. So we have head-fixed mice that are looking at a screen. And on this screen, there is going to be a visual grading that's displayed either on the right side or the left side of the screen. And the goal of the mouse is to move this little wheel that's coupled to the visual stimulus and move the visual stimulus into the center of the screen. And if the mouse does that in under a minute, then it gets a water reward. If it doesn't move the stimulus or it moves it off the screen, then it will get a noise burst and a timeout. So the visual stimuli themselves have varying contrasts. And so this contrast is going to be randomly selected on every single trial. And so the high contrast gradings are very easy. The mice typically perform very well on these high contrast trials. There are also low and no contrast trials. So on these no contrast trials, the mice don't see anything at all on the screen, and they kind of just randomly or somewhat randomly make a decision right or left. And so the general structure of one of these trials is that we impose a quiescence period on the mouse. So the mouse needs to not move the wheel for 400 to 700 milliseconds, and this just kind of ensures that the mouse is ready and it's not, moving the wheel around and paying attention to something else. There's then the stimulant onset. The stimulus appears on the screen and there's a tone. And then the mouse has 60 seconds to make its decision with this wheel. And then there is a feedback period, again, where there's either a reward or a punishment that is delivered. So what makes this task even more interesting is that we actually modulate the probability of the stimulus showing up on the right or the left side. So at the beginning of the session, it's 50-50, but at some point we move into these different blocks. And so these blocks of trials have a random length, but their median is around 50 trials. And within a given block, the probability of being on the left side of the screen is either going to be 20% or 80%. And this is an unqueued, these switches are unqueued, so the mouse doesn't know that this block switch has taken place. And the cool thing is that the mice can learn this structure of the task, even though they're not cued to it, and they can use these changing statistics of the task, especially to help them in these kind of low contrast trials. And so you're going to hear more about that tomorrow, but I just want to give you kind of a flavor of what this task looks like. So the important thing for us in the next couple slides is that we have a set of trials. For each trial, we have a stimulus onset, we have some wheel movements, and then we also have a feedback delivery time. Okay, so this is the rig that the mice are in during these sessions. And so the mice sit in this holder here in the middle. We have a bunch of different sensors. There's the screen in front of the mouse. There are a handful of video cameras. Up here, you can see the micro manipulator that's holding the neuropixels probes. And so as these experiments are taking place, we're collecting a lot of data. And so here's some spike sorted neural activity that you can see from one of the probes going through midbrain and hindbrain regions. And then also now we have these behavioral traces. So up to this point, you guys have been talking a lot about spike sorting and just the neural activity not really in the context of a task. And now we have these additional traces. We know when the mouse is licking, when it's moving its whiskers, when it's turning the wheel, the choice that it's making, et cetera. And so like before, we really want to spend a lot of time before applying fancy models or doing too much with the data, really just try to understand some of the basic functional properties of the neural data given the experimental setup. And so the way that we've done that in the International Brain Lab is to build this interactive data visualization website. And I should say that I'm not the one that built this website. There are a lot of very talented engineers in the IBL that have done this. So just want to give a shout out to them. So this webpage shows a lot of different overview plots of kind of raw data and process data. And so I just want to give you some of the highlights of this. And the homework assignment, which I'll talk about more in a few minutes, is going to be going into this website and just playing around, just clicking around, seeing what the data look like, et cetera. So the first piece of this website up at the very top is the session selector. So you can select a particular session. You can search by the lab or the mouse or the date or the regions that a probe passes through. So there's a lot of ways to select an individual probe or session that you're interested in looking at. So that's something that I'll have you all play around with. The next section of this website is sort of an overview of the ePhys data. So this should be very familiar to all of you now. So there's a raster plot of the spike sorted data. There's some chunks of the raw data down below. There's some histology data in here as well. And so this is really just giving like a big snapshot of what the ePhys data looks like. The next section of this website is a session overview of the behavior. So now we can look at the mouse's performance as a function of stimulus contrast. We can look at reaction times. There are a bunch of behavior traces like from the wheel or from the pose tracking that's aligned to specific events in the trial. And so again, this is all just kind of good sanity checking on the data to make sure that things appear as we expected. And I can say from experience that it's really great to look at things at this level because you often find problems in the data that you didn't think were there. Maybe there's some timestamps that have been misaligned or some pose tracking algorithms that failed. And so it's really good to look at this data as closely as possible before just starting to throw algorithms at it. So below this, the next section is this cool single trial overview. And so here you can go into individual trials in this session, and you can look at rasters of neural data, behavioral traces. There's even a little video here that will show you where the stimulus was on the screen throughout the trial. And it shows you the actual video clips of the mouse during this particular trial. So this is a cool way to get a really fine-grained understanding of what's going on trial by trial. And then finally, the last piece of this website is a single cluster overview for each of the clusters that come out of our spike sorting algorithm. And so this is the one that I want to focus on a little bit more, and this will be something that you all will look at in more depth in the homework assignment. So this is that single cluster overview piece of the website. And so some of this will probably look familiar by now. In the upper left-hand corner, we have all of the good units that have passed the quality control metrics that Chris talked about earlier. And so on the y-axis is the depth along the probe, and you can see information about the brain region that we're in. And then you can also see the amplitude of the spike waveform on the x-axis. And so this is just a static image here, but in the website, you can click around on these individual cells. Whenever you click one of these, it will populate the rest of these plots with information from that particular cluster. So again, some of these you have seen before. Here's the spatial extent of the waveforms. Here are some of the plots that go into computing those QC metrics that you've seen before. And now the new thing are these PSTHs and some other plots that are related, again, to the functional properties of this particular cluster that we're looking at. So for example, here, this is a raster plot of this particular neuron. So we're looking at the times that this neuron is spiked across all of the different trials. And we're aligning, in this case, to the stimulus onset. And here along the side, you can see this helpful color code that tells you which block type that you're in. So if the probability of the stimulus being on the left-hand side is 50% or 20% or 80%. And these PSTHs are colored accordingly. So you can also see similar types of plots for, say, the stimulus contrast. And so for this particular cluster, you can see that it responds with a higher firing rate for these high contrast stimuli and a much lower firing rate for the low contrast stimuli. Similarly, you can see the neurons PSTHs for right versus left side of the screen, and also if the choice was correct or incorrect. And so again, this is a really helpful place to start exploring your data. I think that's probably one of the big take-home messages of this whole workshop is, there are a lot of tools that people are developing to look at the data, and because that's really, really important. And so this is sort of the next step after you've gone through, the raw data, looking at these different spike quality metrics and things like that, just starting to do some basic alignment to trial events and see if you're seeing responses that you would expect. So I think that's all that I have to say about the website. So I'll talk a little bit about the sort of the practical assignment. And I'll post these slides in the Slack and also some more information in the Slack. So what I want you all to do is go to this visualization website that I just showed you. You don't have to install anything, which is nice. At the very top, you can choose different sessions that you want to look at. And there are two main things that I think will be interesting to look at. So one is using that single cluster overview section that was at the bottom of the page that I just described. See if you can find clusters that differentially encode aspects of this particular task. So for example, can you find a cluster that encodes the stimulus side differentially between the left and the right side? And so I've given you some hints here. You can check this particular probe, and you can enter this probe ID into the session selector at the very top. And then you can click around the different clusters and see, is there a cluster in there that responds differently to stimuli that are on the left versus on the right? Similarly, you can find clusters that respond differently to the different block types or to different feedback types. So again, I've given you some hints for some places to start in all of these, but really this is just to kind of encourage you to click around the website and get a sense for the different types of things that you could look at in this type of experiment. And then the second thing that I think is interesting is maybe overlapping a little bit with some of the homework assignments that you did yesterday. So one is, again, just to click around, look at a bunch of different probes. and see if you can find a probe that has relatively stable firing rates over the course of the session. Hopefully this shouldn't be too hard to find. The next one is maybe a little bit trickier, but try to find a session where individual clusters, these spike sorted units, become obviously more or less active over time. And so again, I've given you a hint here that you can go look at to see what I mean by that. And then you can also see if you can find some other ones yourself. And tomorrow, what we'll talk about, or one of the things that we can talk about, is if you find these clusters that seem to be present in the beginning of the session but not the end of the session, or are present in certain epochs but not in other epochs, should you include these clusters in your downstream analyses? So that's something that you all should think about, and we can discuss more tomorrow. Okay, so I'll stop there, and happy to take questions if we have any time.